Gene editing. You've heard about it everywhere, in books, in movies, even in video games. But it wasn't until very recently that scientists started to consider this as an actual possibility. I mean, come on, editing someone's DNA? Changing the code? That's impossible, right? Not as much as you might think. After years of research, scientists have finally discovered a way to make this happen, and that's what I'm going to tell you about today. The magic of gene editing with Crispr Cas9. Crispr Cas9 was inspired by the defense system of bacteria. It is done by cutting out an unwanted strand of DNA and then replacing it with a new and necessary strand. Scientists begin the process of gene editing by injecting a Cas9 protein. These proteins are important for cutting out those unwanted strands. The Cas9 begins its journey equipped with guide RNA, which includes a 20 nucleotide stretch that matches the strand that needs to be cut out. The Cas protein uses this to find the complementary stretch of DNA. It's like if the Cas was a police officer choosing the right criminal out of a lineup. Usually, it'd be hard for the policeman to choose the correct person, but if they had a picture, then this process would be easy. The guide RNA is like that picture. It makes it easy for the Cas to find the right strand. Once it thinks it has the correct strand, the Cas protein will use a PAM sequence to help it verify that it has in fact found what it is looking for. The PAM sequence is a 3 nucleotide stretch of DNA that is found adjacent to the stretch that needs to be cut out. It's used to help the Cas protein verify that it has found the strand that they want to cut out. The PAM sequence works almost like an ID or a driver's license. It helps the police officer know that you are who you say you are. The Cas9 can now make its cut. Now that there is an empty space in the code, the scientist has a chance to insert a new strand of DNA called donor DNA. This gives the cell a choice as to how it will incorporate the new information. It can either pull the strand into its code, or it can use it as a template to rebuild its strand, therefore copying the donor DNA. This is almost like buying a new pillow. You can either just buy the pillow and put it on your window seat, or you can make an exact replica of the pillow and then put it on your window seat. Either way, the pillow ends up on your window seat, but the way it got there is different. The scientist has no control over which method the cell will use, so all they can do is put in the donor DNA and hope for the best. Now, there are a variety of methods that are generally used to insert the DNA, such as using bubbles of fat that will fuse with the membrane or electric currents. The most common method, however, is to use a virus. In this method, the viral DNA is extracted and replaced with donor DNA. Then the virus is simply put on the cell and allowed to inject the DNA as it usually does. While this system is not yet ready to be applied to the real world, it has still helped scientists with a lot of super important research. We have been able to make better models of diseases in animals, and we've been able to figure out the functions of different stretches of DNA. After all, this is just the beginning of our journey with gene editing. Who knows what we could discover next?